Enterobacteriaceae has two major groups. First group comprises those species that colonize the human gastrointestinal, but most associated with human infections. The second group consists of those species that colonize humans, but rarely associated with human infections. So in this video, I will be discussing what are the species under Enterobacteriaceae and what are the tests involved. So as you can see in the diagram, there is an oxidase test that determines the presence of bacterial cytochrome oxidase. It has a negative and positive test. So in negative test, there is no color development and an absence of enzyme. The bacteria found in the negative test is the Enterobacteriaceae. As you can see. So this bacteria is a form of glucose and a catalase positive. So it is a gram-negative and a facultative anaerobes. So some of the species under Enterobacteriaceae that are the causative agent of typhoid fever and dysentery are the Salmonella typhi and Shigella spp. So these organisms have simple nutritional requirements. So a McConkey agar is used to isolate and differentiate organisms of the Entero family. So the colonies on the McConkey will turn into pink if it is a lactose fermenter. So the IMVC test is a group of individual tests that are commonly used to identify bacterial species. So the E. coli can be found in the foods and intestines. So on the other side, below on the MVC test is the motility test. So this test will use to determine whether an organism is a motile or a non-motile. Motility test. So Klebsiella is a non-motile bacteria. So it is straight rods and oxidase negative. So K-pneumoniae is a species under the Klebsiella that can cause hemorrhage in the lung tissue. So Enterobacter is a motile bacteria. So it is straight rods and oxidase negative. So some of the infections of Enterobacter are urinary tract infection, or also known as UTI, and skin and soft tissue infection. So as you can see, the hydrosulfide test has a positive and negative test. So Shigella is the bacteria that reacts to the positive test. This bacteria does not produce a hydrogen sulfide gas, in which the resulting colonies will be colorless. So for the negative test, Proteus and Salmonella are the bacteria that reacts. So there's a test in which the urease test will use to determine the ability of a microorganism to produce enzyme urease. Here. Okay, so the bacteria that reacts to the positive urease test, the Proteus. So it is a rod shape, a gram-positive, and catalase positive. So for the negative urease test, the bacteria that reacts in the Salmonella, in which the urea is not hydrolyzed, is the Salmonella. So it has the ability to invade or survive in human host cells, resulting in fatal diseases. So I will now move on to the bacteria that reacts to the positive oxidase test. So Pseudomonas is slender, broad-shaped bacteria. So it is a motile bacterium, and P. aeruginosa is one of the species of Pseudomonas. So it is an opportunistic pathogen. So the bacteria will invade the immune system of an individual, uh, in which it will get weakened and will create an infection. So that's all for this video, and thank you.